Today, we're delighted to invite Thomas Wingo, a neurologist from Emory University, who is also leading a biomarker core in the Emory ADRC. He has done a lot of his training at Emory. It sounds like essentially did his MD at Emory and then followed it up with internship and neurology residency at Emory, followed by a cognitive and behavioral fellowship under James Law, and then subsequently a postdoc fellowship in translational neurology and population genetics. And that really leads into the set of tremendous publications and grant awards that Dr. Wingo has been a recipient of and author of publications in looking at genetic risk factors for Alzheimer's disease and the associated proteome with that risk. It's really innovative to kind of now use the latest tools in genetic studies to be able to now combine across multiple factors and kind of get what might be a polygenic risk score or other pieces that a number of groups have been doing. And I think Dr. Wingo has been adding that with Proteomes. So we're really excited to hear what he has to say. So Dr. Wingo, we really appreciate you coming today and look forward to your talk.
Well, thank you very much, Dr. Brewer, for that kind introduction. As already alluded to, I am a neurologist by training, and I also consider myself a human geneticist. And I'm going to talk to you today about some of our work integrating and understanding GWAS and how brain proteomes can inform us finding new causes for Alzheimer's disease and other brain illnesses. By way of disclosure, I have an affiliation or a co-founder of a small biotech company that has no role in this talk. So just to frame, since I never know precisely who's going to be in the audience, I want to make sure we're all starting at the same spot. And that's why I just put up the central dogma here. So the DNA to RNA to, DNA to RNA to protein. And then really as a neurologist, a cognitive neurologist, and somebody that's interested in the brain, as you all are very well aware, I'm sure, that the brain illnesses, the neurodegenerative ones especially, are often defined by protein accumulations or other protein changes. And so it's been my focus to really try to understand the relationship between protein and disease, here illustrated between these two brains. One is a 100-year-old woman that died with normal cognition, and this is a 65-year-old man who obviously has a lot of mediotemporal lobe atrophy and had a syndrome of Alzheimer's disease, dementia in life. But the story is not that simple, and so because disease itself can cause changes to protein abundance, and also proteins are regulated. So there's a complex relationship between proteins and messenger RNA. And so it's not straightforward for us to be able to interpret at the time of death, for instance, how proteins are influencing disease, especially just looking at their abundance. And so our work is really aimed at trying to understand or deconvolve that relationship or reduce some of the complexity to identify what we can find from that relationship. And so the way we're going to do that, and to prelude the entire talk, is by looking at how does the genetically controlled portion of protein abundance predispose to disease. And so how are we going to do that? The first, we're going to recognize that while there's a lot of complexity in the relationship between protein and disease and RNA and protein abundance, for instance, one thing that we can feel fairly confident about is that DNA variation is not something that's going to be influenced by disease. So the direction is going to be DNA variation will contribute to disease, but not by and large the other way around. And by that same token, DNA variation may contribute to protein abundance, but protein abundance will not, by and large, contribute to new DNA variation, especially at the population level, which is where we're going to really be examining it. And so we can use the fact that we have these two genetic relationships, DNA to protein, DNA to trait, to resolve this relationship between protein abundance, or the inherited factors that may contribute to protein abundance, and their relationship with disease, to infer what may be putatively causal, or in my word, I mean, and that's what the words in the community, in my view, these are putatively predisposing to disease, in my opinion. And so how are we going to do that? Well, the first relationship, what I told you, was so DNA to trait. And so that is, we've learned about that through the era of GWAS. And so I think many people are intimately familiar with this plot, the so-called Manhattan plot, but let's just go over it briefly to make sure we're all on the same page. What we have is, on the x-axis, we have individual sites of variation within the genome. And these are little dots, and sometimes they're hard to see because they're all overplotted or smooshed together because there's so many of them. But you can see a few individual ones. And these dots are ordered in their chromosomal position. And we have the chromosomes 1 through 22, which is not labeled here. And then, of course, most GWAS, sad to say, do not handle the X or Y chromosome. Particularly X they're hard chromosomes to analyze, so there may be some reasons for that. But what we're focusing on here are autosomes, and on their on common variants. So these are variation in the population that occurs at or above 1% minor allele frequency. So out of 100 people, one person has a minor allele or is a heterozygote for some of those changes. And so those are the dots. And then what we're plotting is the negative log p-value. So this is just the strength of association for the in this case, the test statistic is probably a logistic regression. So this happens to be a Manhattan plot from AD/GWAS. And so we have a few lines that are helpfully drawn. And basically, the idea is that if you perform 1 million tests by the central limit theorem, you're going to inevitably find some tests that are by chance really significantly associated with your outcome. And you need to account for that. You don't want to chase around or chase down things that are simply due to chance. So we artificially, or we impose a restriction on this sort of a significance level that we expect, or we want to say something is significant. So that's what these peaks right here are colored by in blue. And the idea is that of these peaks is that these are, and the reason there is a peak is that all these markers that are in this little region are all linked together. And so let's just take a look at a few examples. And so we have the ApoE locus, the venerated locus in Alzheimer's disease, and then we have these loci down here that are a little bit less strongly associated. And so if we look inside this locus, so remember before they had labeled the locus as ApoE, and I just want to use this time to share with you that there are a lot of genes within this locus. ApoE is clearly incontrovertibly associated with Alzheimer's disease. The axis here is to the negative log of 300. So that's about as close to zero as really is necessary for these kind of analyses. And you see they're all topped out right there too. And ApoE is really the most likely causal gene here. If there were other causes, it'd be very difficult to detect. But the point is that it has 2 known coding variants and the cysteine to arginine changes that are incontrovertibly associated with Alzheimer's disease. By contrast, there are many sites that are a little bit more like this, where we have a lead site. So this is one area of genetic variation that is associated with the disease. Or the outcome of Alzheimer's disease. And then there are other sites that are sort of associated or physically linked to this site. So they have an elevated association by virtue of their physical linkage with the site. And it's really unclear what this site really refers to or why it's associated. And there are a lot of genes in the region, so resolving that is important. And I just named 2. FUS is obviously associated with FTD, and STX4 will come a little bit later in the talk. And the point of what I was trying to share with you is that we're able to use the era of GWAS, the results of GWAS, to understand something about how DNA is associated, or genetic variants within DNA, pardon me, are associated with Alzheimer's disease. And so that completes one of our sort of legs of the triangle that we're going to try to build. The next is to try to understand how our proteins and DNA are related to one another, and how specifically how protein abundance is. And so I guess I already alluded to why I study the proteome, but it bears some maybe explicit description that the proteins really are obviously the hallmarks of neurodegenerative diseases. They're really the cellular, executor of the will of the cells. They often are extracellular. And there's not a perfect correlation between messenger RNA and proteins. And especially at the end of life, we find the correlation, the median correlation is in the low teens between the messenger RNA and protein abundance. So they're not great surrogates for one another. And the other nice thing about proteins is they're relatively stable, and they are often the biomarkers and drug targets. So they're a little bit closer to what we are wanting to understand, in my opinion, for understanding the mechanisms of disease. And so how do we study the proteome? Well, we use mass spectrometry-based proteomics, and this is but one of the several methods that one can employ. And so I just thought it would be worth pointing out how, in general, this approach works. We have a protein mixture represented here by these squiggly lines with blue and other color right here that we're going to sort of follow through. And what we do is take the protein mixture, which has been isolated from the human brain, and we then digest it, and represented by Pac-Man. And we use the digestion primarily to sort of reduce the complexity of these peptides and make them more analyzable. And then we are going to separate these peptides based on their biophysical properties using HPLC. And that allows us to sort of enable better detection of the peptides. And we send it through a mass spectrometer, and we have a couple different scans, one to sort of identify initially the peptide, and then two to fragment the peptide itself. And so the fragmented peptide, these little lines to a person that does mass spectrometry mean a lot. And what we can infer from these lines, and they're really ionization strength of different ions, we can use a database of the human proteome, and we can basically ask how close would this pattern match an existing putative pattern within the database. So it's a database lookup sort of approach to mass spectrometry. And this is commonly called a sort of bottom-up proteomics. We're identifying particular fragment peptides, and then we're able to map those peptides back to proteins. And we're able to integrate the area under the curve, essentially, of their ion intensity to infer something about their protein abundance within the sample. And typically, we're able to analyze about 9,000 proteins from human brain using a variety of strategies, but this is the general overview. And so how are we going to infer something about protein abundance in the brain at the time of death or after the person has died, and how their genetic variation? So what we're going to do is something called QTL mapping. So that is a quantitative trait locus mapping is the other way of calling it. And basically, we have proteins, and there's some natural variation that occurs in the population of these proteins that we observe. And then we're going to ask, how does the protein abundance associate with individual single nucleotide variants or other variants that occur naturally in the human population? So this is a cartoon diagram of a gene. So we have the coding region right here, and then these are sites of natural variation. And we can ask individually, does this site this site associate with protein abundance, and does this site associate with protein abundance, and so forth? Or we could do something a little fancier and ask, like, collectively, how well do these sites predict protein abundance? So both of these approaches are used, but the essence that I want you to take away, or the the takeaway point is that we can infer something about protein, about the relationship between DNA and protein abundance, and that's what we're doing here. And so one of the really interesting facets of this work led us to identify that actually the genetic regulation of proteins, there is a significant portion that is distinct from transcripts. So as you can imagine, and you undoubtedly heard about, many people have done this for transcripts before proteins were really measured widely or at large scale to really perform those sort of genetic association testing. And we compared the effect of the same site in protein shown on the y-axis, and on the left, on the x-axis, we have the association of that same site with messenger RNA. And I'll draw your attention to the blue area right here, which is a site that essentially has very little influence on the RNA abundance, but it has a fairly strong influence on the protein abundance, either up or down. And these are a minority of sites, but they're an important number of sites, about 40%, and that are what are called PQTLs, or protein quantitative trait loci. That's what these individual sites are called, and I'll use that terminology throughout the rest of the talk. So about 40% of those PQTLs have no corresponding EQTL that is discernible. And I think that's very fascinating. It certainly implies, or it leads to possibilities to understand things like post-translational effects and so forth. And it also highlights that proteins and transcripts are distinct entities that undergo distinct regulation. And but turning now back to our original sort of motivator. So we have now what we need to start to address this relationship, the genetically controlled portion of protein abundance and its relationship with disease. And so the next thing that so to say it sort of slightly differently, what we have were the genetic variants, and we're going to ask, how are those identified or associated with AD? So that's what we get from GWAS. And then we're going to use how those same genetic variants influence brain protein abundance and ask, and that's how we get that from QTL. And so we're going to ask ourselves, does the genetic variance association with AD, is it acting through that of brain protein abundance level? And essentially, the first pass of this, we're going to reweight the GWAS results by the site's influence on brain QTLs, on brain protein QTLs. And so we're going to essentially resolve some of these loci into specific proteins. And that's what's shown on the right-hand panel, the sort of result of that. Before I delve into the details, though, I would be remiss if I didn't acknowledge the incredible and selfless contribution of those people who were, when they were done with their brain, they donated it to science, and they come from or were participating in several studies, in the Religious Order Study and Memory and Aging Project run by David Bennett, and that is our discovery data set in this study, and then the replication data set from Banner's Sun Health Systems with through collaboration with Tom Beach and Eric Riemann. And we examined the dorsolateral prefrontal cortex. The particular reason this was chosen was mainly because of the that it's not severely affected in later illness, so we could still glean information from the tissue, even if the person had maybe more advanced Alzheimer's disease, dementia, and neuropathology. So, the result of the integration is shown sort of here on the left, and then the table right here gives you the actual numbers. And what we find are 12 or so proteins that are strongly implicated in that inherited genetic variation that influences protein abundance, at least is plausibly related to the development of the outcome in this case, Alzheimer's disease. And the first thing after you get a result is you want to verify it or have some other approach to see if there's consistency within data set or within the data. And we don't have an independent GWAS. So this really is just are limited in that way. But we can use the same GWAS for both the discovery and the confirmatory PWAS, which is the other data set that I was referring to. It is fewer people, so we have less power, but we can ask ourselves, is there consistency between the two data sets? And indeed, we find fairly good consistency within the bounds of reason. There are 4 genes and protein pairs that we're not able to either identify, or they weren't sufficiently heritable in the confirmatory PWAS to really provide us to allow them to go forward through the integration. The next piece that I'll share with you is to really address or try to address the issue of potential causality. And here, when I refer to causality, I'm again referring to inheriting something that is predisposing to disease. And it's more of a statistical way of referring to causality in the literature. But think about, these are variants that are being inherited that contribute to disease risk through their effect on protein abundance. And one of the approaches that we use to try to get at this notion of whether these sites or these protein and gene pairs were potentially causal was whether they co-localized, and using a software called Coloc, which basically, I'll summarize for you by illustrating that it's a Bayesian approach. So it tests several hypotheses, and the H4 is probably the most interesting for our purpose. And it's basically asking, do the sites that influence or are associated with the outcome, are they the same sites that are associated with the protein abundance? And we see that for about almost 10 of those genes they are, and then there's some that we don't have sufficient number of sites within the gene to really make that causal or draw that conclusion. And then we have some that are clearly not. And the reasons they aren't overlapping is probably due to linkage disequilibrium, which I sort of alluded to before, but it's the correlation among markers. And so what I would consider this to be, these correlation among markers is really not what we're interested in. And so I think that excludes or helps sort of lower down their probability of those being actually causal. And then the second approach that we used was really to look at or to test a very similar idea. And I'll just share with you the intuition. And it's using a software and an approach called Sunbury statistic Mendelian randomization. The idea is that if you think a protein is contributing to the disease, and it's inherited in a way that contributes to the disease, and that if, let's say, higher protein abundance contributes to higher disease risk, then you would expect that there should be a relationship between the effect of the strength of a QTL, that is the strength of the protein quantitative trait loci, and the effect of that site on its association with Alzheimer's disease. And that's what's illustrated in this plot on the left. And this is from Xu et al.'s Nature Genetics paper that really described the method. But the idea is that the stronger the effect of the GWAS, so the higher on this axis is GWAS, is the beta value of GWAS, or the strength of association with the GWAS. This axis is the effect on QTL for the same site. So these are paired sites. So this is a site that is contributing to both. And what they have, the crosshairs is the error measurements of these things. But the overall point is that there is a relationship, this regression line that can be drawn. And that would imply that there is evidence for causality among these sites on the protein abundance, that the protein abundance is actually somehow influencing the inherited risk for the disease. And so we applied that same approach, and we find that, and the real sort of column to pay attention to initially is this p-value column. So there's evidence for many sites that are consistent with that, this idea that there is some relationship between the effect on the GWAS of the site and the effect of the pQTL in this case. We also applied something called this Heidi outlier value test. So this is basically asking if there is consistency among all the sites, and that whether this may be this value is really driven by an outlier. And in this case, for instance, EPHX2, it's really associated with an outlier, and so we would downrank that. And so for myself and for everybody else, I try to make it a little easier by just putting this final column that says evidence for causality. So this one is lower evidence for causality, same with this one and ACE. And then this one didn't have enough variance, and this one didn't have enough variance for us to be able to move forward with the analysis. So, so to summarize this portion of the results, we have 4 genes that are really consistent in both the discovery, replication, PWAS, the colocalization analysis, and from the SMR. So these are measures of evidence for causality, essentially. Then we have 4 genes that are, and they're protein pairs, that have sort of a mixed picture, where there's some evidence for, or there's evidence for, in both discovery and replication, but then coloc and SMR sort of disagree a little, to some degree, about the strength of the causal inference that can be made. And then we have another class where there is evidence for causality in the discovery, but we don't have enough data to move forward with the replication, because the replication set either didn't measure them or there just weren't sufficient number of people. And that's a problem with proteomics, that you don't always have the same precise coverage, and that sort of distinguishes it from transcriptomic data, unfortunately. But then also these have evidence for causality. So I would place them sort of in a middle ground. And so the next, moving from those results, the next question that I think comes to my mind, and probably many other people, is whether there's evidence at the transcriptomic level for those things. Since I told you before, there's not a lot of evidence that brain protein and transcripts are really highly correlated, but the genetic portion of the regulation of brain transcriptome may be correlated. And so that was what we tested next. So we used the same pipeline that I already described for Alzheimer's disease and the proteomes, with these brain transcriptomes from mostly the same data sets, or the same participant donors who had their brain proteomes also measured in the dorsolateral prefrontal cortex. And so what I would draw your eye to is this final column. And when we look at it, this is the 4 genes that had really evidence across the board. And interestingly enough, so the big plus here means that it was an FDR significant. So I should back up by saying all these were FDR, adjusted for multiple testing comparison. But this one in the TWAS context was significant. This was only nominally associated. And then these, either the DOC2A was not a transcript that was measured, and then this one had no evidence for association. So about half have this one have evidence. At this level, there is evidence for consistency, probably a little weaker for CTSH. And then finally, there is evidence for STX4. So the way I take this is, the way I interpret this is, this transcript was completely different technology. It's the same brains, but it's completely different technology that is assigning abundance values to these. And so it certainly is very supportive of the initial proteomic findings. And so that gave us some confidence that in these results. So to summarize this sort of first portion of the talk, I hope I've shared with you that we can integrate protein expression. We can the genetic regulation of protein expression with AD/GWAS results to identify and refine common genetic variants that are associated with AD. And in case it wasn't apparent, there were 3 genes that were most proximal to the AD/GWAS loci. So when people do GWAS, they often just take the lead SNP, or the most strongly associated SNP in a region, and they just label it with the gene's name, which is a completely reasonable thing to do. And it happens that we find evidence for 3 of those are really associated through the proteome. And then 11 genes that we identify were not the most proximal to the AD/GWAS loci. And then we have a ranking of 4 genes with much stronger evidence and 3 that are of high interest, but maybe not the strongest evidence. One thing I didn't go through, but I just thought I would share, is that most of these tend to be expressed in neurons, at least through using single cell sequencing data. So with that, I'd like to shift gears slightly. And since that work was really motivating and motivated us to really want to explore the shared contributions to the pathogenesis of psychiatric and neurodegenerative diseases. And this is really working in collaboration with my lab and life partner, Aliza Wingo, who is a psychiatrist and geneticist. And so, this is really born out of our sort of ended up being accidental collaboration over sort of shared interest on this topic. And so most people know this already, but it just bears orienting us to the same page that typically psychiatric illnesses either occur earlier in life or early to midlife, and they have echoing ramifications on late life illnesses like Lewy body disease and Alzheimer's dementia. And epidemiologically, this is borne out in evidence, at least for schizophrenia, that they have increased risk of developing dementia at the end of life, not maybe what I would consider, or many consider, Alzheimer's per se, but some form of dementia. And that people that have midlife depression also have an increased risk of developing dementia. And interestingly, as I would imagine, everybody that sees patients that have dementia and Alzheimer's disease especially, that most patients, and this is a study that says 80%, although my personal view is this is higher, have some degree of neuropsychiatric symptoms if you have dementia. And so there's a curious relationship in terms of this phenomenology that we wanted to explore and really ask, is there some sort of genetic or molecular connection that we can identify? And so how do we approach this? So we used a lot of the tools that I shared with you before and sort of illustrated before. And the reason I illustrated with them was because I'm going to echo them right here. And we looked at very large genome-wide association studies and that were of psychiatric conditions or neurodegenerative conditions. And we first asked a very basic question among those using that GWAS result. Do you find any evidence for a shared genetic basis among psychiatric diseases and those that are late life dementing illnesses or neurodegenerative diseases? And so Alzheimer's disease, really, I would say this is probably better described as dementia, since the GWAS that we used was included individuals who just had they answered a affirmative or negative based on, does my parent have a family? Did my parent have dementia? So really a much broader phenotype, in my opinion, than Alzheimer's disease. But I think Alzheimer's disease is probably by far and away the most common cause of dementia. So so that's why we use this shorthand here. But I do acknowledge that it's probably a mixed bag. But the point really is that even among or using that data, we see that there's an interesting genetic correlations. And so how do we do that? We used something called LD score regression that asks whether accounting for things like linkage disequilibrium between the samples, is there some shared genetic architecture between these conditions? And for an example, Alzheimer's disease, we see significant sharing with major depression. It's certainly echoed or consistent with the epidemiologic literature and a lot of other psychiatric conditions as well. Lewy body dementia, surprisingly, or maybe unsurprisingly for some, is associated only with anxiety. And I was surprised by amyotrophic lateral sclerosis and its strong association with anxiety. Right now, and so just to share with you guys that, so in this work, we also looked at brain volume, since I thought there are some people that are very interested in that among the crowd. and MRI measures of volumetrics and things like that. And clearly, and this is maybe a little hard to see, and I apologize for that. I'm really not sure how else I could have done it. What I wanted to draw your attention to, so we have these two Alzheimer's studies they called AD1 and 2. This is really the dementia one. This is really of clinical Alzheimer's disease. I just wanted to point out that there is some association with Alzheimer's disease and hippocampal volume in an inverse relationship. Certainly that would make some sense. Curiously, cortical surface area is associated with many psychiatric conditions, and that I wasn't quite expecting. And so the view is, or the view that I'm coming to, is that there's a lot of sharing among brain illnesses, as one might expect. The sharing that you see is, among the structural imaging is mostly among itself. Among the neurodegenerative, the psychiatric disease, it's mostly among itself. And then this neurodegenerative diseases are shared surprisingly amount is right here with some of the common psychiatric disorders, leading us to want to go a little further and understand whether the potential shared genetic mechanisms and shared proteins that may be involved in these conditions. And so the way we did that was compared to the last study. We have a data set of now 720 individuals with brain proteomes. And we used the GWAS that I just described, psychiatric, neurodegenerative. And we performed the same sort of integration. And I may be remiss in failing to say that the integration, the sort of first step of integration that we did, is often called PWAS, or proteome-wide association study, which follows along TWAS, which is the transcriptome-wide association study. And again, it's really looking at the genetically regulated portion of protein abundance and whether that's associated with the outcome. And that's what is sort of shown in this this diagram right here. We're interested in genes and their genetic variants that are acting through brain protein level to contribute to disease, and that is explaining why those genetic variances are associated with disease in the first place. So what are the results of that study? So this is a little bit of a busy diagram, and so let me just walk us through it. Right at the first step, I'd just like to point out the causal proteins that we identified. So neuroticism 129 or 24, major depression 98, and schizophrenia 158, and say Alzheimer's disease. This is the more dementia leaning Alzheimer's disease 18. What I also want to point out to you is that we have a percentage of sharing, and then either it's shared within a group, and that means either within psychiatric or within neurodegenerative, and then we'll get to the shared between groups in a moment. What I thought was interesting, and I kind of expected, to be frank with you, was that psychiatric illness within the group, there's a lot of sharing. So you see the gray bar here, 35% of these 124 proteins are also identified in collectively these other conditions. And you see a modest degree of sharing between, say, PD and AD within the other group. Obviously, ALS did not have very many shared. And then Kunkel's 4, I wouldn't read too much into this percent sharing, since it's really shared with this AD, the dementia AD one. So that implies that between or within groups, there's a lot of sharing. And so one way we can think about why is there sharing, and one of the ones that I've just sort of talked to you about, is, do we identify the same protein? So in here is a sort of a diagram of, if we have a given protein, does it share in between different diseases? So like maybe this is one disease and this is another and this is another. The other way we can think about shared mechanisms is whether the proteins that we identify with one condition are also physically interacting with proteins that are implicated in others. And we've explored both of those. So the first thing I would just point out is that between groups, we see a little bit more sharing among the neurodegenerative diseases compared to or with the psychiatric versus the psychiatric to the neurodegenerative, which I thought was interesting and certainly spoke to maybe the risk factor for some of these conditions being risk factors for this later life conditions. And just to give you a few examples of sort of what we saw, especially among the shared causal proteins, we have STX1B, for instance, implicated in neuroticism and in anxiety, and appears to be shared with, also implicated in Parkinson's disease and in dementia. We have MTSS1L that is shared or identified in bipolar and in AD, and shared among those two. And then we have DOC2A and CCDC6, and these are implicated in schizophrenia and in bipolar, then also shared with Alzheimer's disease. And then finally, ADAM10 and ACE, implicated, interestingly enough, in schizophrenia and also in Alzheimer's disease. So that's one way of examining the sharing. And so the second way is really asking ourselves whether those proteins that we found collectively, do they share some physical interactions with one another? And the way we did that was looking at a curated list of from BioGrid, which I acknowledge, is it's a large swath of data, but we did select out the ones that have evidence for direct protein-protein interaction. And we found that there was 2.6 times more protein-protein interactions between psychiatric and neurodegenerative diseases of the causal proteins that we identified for both than by chance alone. And here's an example of what that looks like. So we plotted neurodegenerative disease is in this color, and I lack trichromatic color vision, so I'm not precisely sure what color this is, but this is, I think, an orange color. And so you see there's a contrast between the two of them. And we, for instance, find LACT-B has a number of connections with 10 other psychiatric causal proteins, which I think is kind of interesting. We also see STX6 implicated in AD, and has interactions with 7 other proteins that are implicated in other psychiatric diseases. And finally, CCDC6 is itself an AD causal protein implicated in a psychiatric condition, and it interacts with 7 other proteins that are implicated in psychiatric conditions. To provide a little bit more granularity around that, we have the different conditions listed out, and certainly I would encourage you to reach out to me or follow up in the paper to find out if there's any of these that are interesting to you. But I find that the variety that are of conditions that are associating through different proteins very fascinating. And so with that, since we're approaching the end of our time together, I wanted to summarize this section and by saying that overall, I hope you would walk away with thinking that there is a shared genetic basis between psychiatric and neurodegenerative diseases, especially really using common variants. That's what we really explored here. And then that there is evidence for a shared genetic diathesis between psychiatric and neurodegenerative diseases that are manifested by causal brain proteins themselves, and that there's really evidence for enrichment among these shared causal brain proteins for psychiatric and neurodegenerative diseases. And so I thought I would share some of our ongoing work that and sort of directions that we're taking to move this further, since this is just the initial foray into it and certainly not a final story. And the first is that we have spent, I guess, 1.5 years or so trying to understand the role of sex on brain transcript and protein expression and the genetic regulation thereof, and have recently submitted a paper for publication on that, and it's very exciting, and it certainly resolves certain PQTLs down to specific cell types and specific enhancers that I think would certainly be very relevant for disease and understanding disease causation and pathogenesis, and why there may be risk differences between sex. And then we also are actively working with partners to try to validate some of these causal proteins in models of human illness and brain illness. And then we're also working on a project to identify individual peptide risk using whole genome sequencing coupled with the mass spec-based proteomics, where we can identify peptide variants that are associated with disease risk that are maybe coding. And then we're expanding the brain proteomics to include people of diverse backgrounds. I'm remiss in failing to say that all of the work that we did thus far really focuses on people of white European ancestry, and we certainly need to do a better job of being inclusive. And there are efforts to expand that and the brain proteomics especially into people of more diverse ancestry, and we're a part of that with the AMP-AD consortium. And with that, I'd like to thank my wonderful lab mates, my lab and life partner, Eliza Wingo, Kate, Jackie, Selena, Yue, and we all worked on this together, and they're a wonderful collaborative group of individuals. And I feel very lucky to be working with them. And of course, I would be remiss if I didn't acknowledge the tremendous generosity of the donors and their study partners, and being so generous of their selves and their time, and ultimately their brains, and the wonderful collaborators that we have here at Emory and elsewhere, and the funding sources. And with that, I would be glad to take any questions.